Hi, my name is Christine Stanley. I'm the Chief Director of Clinical Genomics at Variantyx. I have 20 years experience in clinical diagnostic testing at multiple molecular diagnostic laboratories, using many different platform methodologies to arrive at diagnoses for patients. The title of my presentation is Unraveling Complex Ataxia Cases Through Whole Genome Sequencing. And this is for the National Ataxia Foundation 64th Annual Ataxia Conference. My disclosures are that I work for two clinical diagnostic testing labs, Variantyx and C2I Genomics. For today's talk, I'm going to give you the overview here. So what we hope to accomplish is to give you the idea about genomes and what they can do. So genomes provide a data platform for comprehensive genetic testing. They allow us to look at unique genetic changes that cause ataxia. And these changes require a unique platform in order to identify them. So we will talk through that. And then we'll go through some cases, some interesting cases that have been solved by genome testing at Variantyx in the area of ataxia. So let's just start with genomes, and we know that genomes provide a massive amount of data, more data than any other genetic test available. But it's more than just the amount of data that's provided by genomes that we harness in order to identify different variant types that cause genetic disease, and I'll walk through what that is. But first we can look at just genetic tests in general. So genetic tests are all start at the same place. They all start with isolated DNA, and then taking that DNA and chopping it up into fragments or pieces. And that's where it begins. And then we see a divergence of different tests and what they do and what part of that DNA is used in an alternative testing platform. So if we look at the group of Legos here as an example of our DNA, our fragmented DNA, if we look at it, genomes will look at everything or try to look at almost everything. So 98% of that fragmented DNA, genomes will sequence. And then other testing on the left side, when you're talking about typical exomes, gene panels, or other lab tests, it takes that fragmented DNA and pulls out certain parts or pieces of it in order to run the assay. And it's the process of taking out the pieces that are needed and then discarded the rest that makes genomes very, very different from other testing methodologies. And if you try to think about it, if you were given 30 seconds to grab out of that pile 10 yellows, reds, greens, blues, oranges, and purples, blacks, grays, whatever, and tried to get an equal amount of each color type, you probably wouldn't come up with an exact number for each color. So that's what the fragmented DNA is like as well. When you try to pull out certain DNA that you're choosing to sequence, you're not going to pull out exactly the same amount of each piece that you're looking for. So when you do that mechanical process of sequencing where you're only looking at a fraction of the total, you're going to have a biased starting set that you sequence, and so your data set will already be skewed a little bit. And that is the issue with trying to extrapolate all these different variant types that you need to look at for genetic testing from methodology that starts with skewing. And genomes, PCR-free genome sequencing, which is what we do at Variantyx, makes a very even covered data set, and that allows us at Variantyx to design software tools that can allow us to look for more difficult to see information, and I'll talk about that in a minute. So when we talk about the tests, again, just to reiterate, genomes gives us the most data, and then exomes, and then panels, or any other kind of smaller test. What you should know is when you pull out that information at the beginning and you're taking out, say, for exomes, all the coding exons, you're discarding all the rest of it. And so once that's gone, it's gone. You in order to get that back, you'd have to go back at the beginning and get the patient's DNA, blood sample or saliva sample, whatever, and extract the DNA and start again to get that other information. But with genomes, it's already there. So even if you choose to look at a subset of the information, all the rest of the information is there, and you can then look at more if you desire later on. That evenness of coverage that I was talking about is represented in this slide. So if we look on the right panel, this is a view of, in IGV, Integrated Genome Viewer, a view of sequencing reads that are lined up to a reference sequence. And what you can notice is the two main things. At the bottom here, you'll see it's the same scale. It's a little bit fuzzy, but it's the same gene, and we're looking at a small one single gene out of the entire sequence, just to visualize here. And if you look at the genome, you'll see that all the reads that are lined up against the reference sequence are here shown, and you don't see any white spaces. So the exons down here would be what an exome is targeting and pulling out, and then it would be sequencing it. And so you can see, if you look on the exome side, that's exactly what you get. Wherever you see an exon here, one of these blue boxes, you see read pileups at those locations, but you don't see any sequence in between because your test isn't designed to capture the DNA pieces that are in between coding sequences. And you might say, well, I only want to see coding sequences. I only want to see expressed genes, which is fine. But the tools that we need to extrapolate other things, it's important to have this information that's in between. And we'll talk about that a little bit later. And then the other thing is, if you say got more of like this region here and less of this region, so you can see these curves at the top show you about how deep the sequencing is in the region, you'll have like smaller peaks and larger peaks. And when you try to look for if there's missing pieces or not, it really is the ratio of peaks to one another that is used to determine if there's any differences in like a deletion or duplication. And so here, where the peak is smaller, your software is going to be challenged with seeing this as a deletion, or whether this just was less covered than some of the other regions, because these exons that are being sequenced in exomes are like the Legos. If you pulled out more reds and less yellows, you're going to get more red sequence than yellow sequence. And so you'll get more of a wave kind of coverage rather than a more of a flat line coverage that you see in genomes. And so what Variantyx did was take that genomic sequence and designed software on top of it to be able to detect variants that aren't normally easily detectable on standardized next generation sequencing tests. And so this was published in BMC Genomics, and we have a diagnostic console that allows us to interrogate many different variant types using this software. And what are the variant types? So this is an ataxia meeting, so we're going to talk about primarily ataxia disorders. And for ataxias, there's specifically a specific variant type that's very important for identifying the cause of disease in patients who have ataxia. So when we look at the important critical variant types, we're going to want small sequence changes, and small sequence changes are available to detect from multiple different types of testing. You can detect it with Sanger sequencing, with single gene panel or exome next gen sequencing assays. You'll get your small sequence changes in the coding regions. And then but there are two really important other variant types for ataxia disorders, and one of them is called structural variants. So that will be larger changes where you have dosage issues. So you might be missing a piece of a gene, or you might have an extra piece of a gene, and that would be part of the structural variant that we would want to be looking at for identifying the cause of disease. And then the most significant alternative variant type that you see in patients who have ataxia is something called tandem repeat expansions, or short tandem repeat expansions, or sometimes you hear trinucleotide repeat expansions. And that is, for trinucleotide, it will be bases of 3, and you'll see that they there's a part of the gene that's repetitive for that sequence. And when that sequence, that repetitive sequence, gets to be large enough size, it can cause the gene not to be made properly, not to be made at all. You know, there's functional consequences of expanding that repetitive region too far. And most of our repetitive regions are bases of 3, but they can be bases of 4, bases of 6, bases of 12. And so we're really just looking at short stretches of repetitive regions, and these are very critical to identify for patients with ataxia. And this is a big shortcoming of other methodologies, the ability to detect structural variants and the ability to detect tandem repeat expansions. So if we look at some ataxias to look at the variant types that are in them, we see that DERP is caused by a short tandem repeat expansion. Episodic ataxias are typically reported as having single nucleotide changes or structural variants. Friedreich ataxia is single nucleotide variant, structural variant, or trinucleotide repeat expansions. And spastic ataxias can be from single nucleotide variants and structural variants. And the Spinocerebellar ataxias are primarily repeat expansions, but can also be caused by these other 2 variants as well. So you can see that for ataxias, it's very important to have those repetitive sequences identified, but it's also important to be able to identify sort of the standard single nucleotide variants, which is that sort of A, C, G, T changes that can occur at the base level. And also it's important to be able to identify structural variants to really get that diagnosis of ataxia in some individuals. So basically, Variantyx has designed a tool to be able to identify all variant types and do that with a single test. Now, if you wanted to identify the other variant types, there are tests out there that you can be used to identify them, but they're not consolidated in one testing platform. And you would have to order multiple methodology tests to get that result. And sometimes you'd have to order them from different clinical laboratories, because not all labs have all the tests, all the genes, and all the platforms for diagnosing patients with ataxia. So what Variantyx did then is unify all the different testing method platforms into one method platform for one unified result. So when we're talking about large structural variants, that's typically performed as a chromosomal microarray, but that chromosomal microarray has limitations to it, where it can't really identify things that are really small, like on the exon level. So each one of these methodologies, even though these are the current, or these have been in the past, the methodologies used for detecting these types of variants, each one has its own limitations associated with it. So if you needed to detect smaller exon level deletions, you would, and duplications, you would order something called MLPA. But not all genes have an MLPA test that have been developed for them, so there's limitations there. There's single nucleotide variants, which you typically are going to pick up on single gene tests, gene panel tests, and exome tests, and genome tests. And then for short tandem repeats, these are typically done by PCR assays or Southern blots. So you see there's multiple method platforms that are out there in the industry right now that are being used to identify these variants, but it's very disjointed. And what Variantyx did was use genomes to identify all of those variant types. What's important about that is if you did have multiple assays that you had to run, you might get half the answer on each assay and get 2 separate reports where you don't have those results pulled together to really understand that you actually have a positive result. So to really kind of bring home this message, I'm going to walk you through some of our patient test results, so you can see how being able to identify these different variant types is very useful for our ability to really provide that cohesive single test result for you. If we think about a typical patient with rare disease, we know that they go through a long diagnostic odyssey. And then part of that is because of all of the different types of tests that are out there, and that there's, you know, different genes and panels and methodologies at different laboratories. And so you would get a test, and then if it's negative, then you come back when the result's back, and then you have a discussion about another test, and that gets ordered, and you get this cycle of like negative results that keep coming back. And so a typical patient will take over 7 years to be diagnosed and have visited 8 different physicians, spending on average $28,000. So there's a lot of cost, time, and probably and frustration involved in not getting to the answer soon enough. So if we look at our first case, this sort of underscores somebody with a longer diagnosis and multiple tests. So this is a 17-year-old male who had a 12-year-old diagnostic odyssey. He was healthy at birth, had a negative family history for any genetic disorders, had excellent upper body strength, but started to have some issues when he was 5 with some leg pain. It was kind of written off, and then between 5 and 12 really didn't hear much from them, but by 12 was actually having quite an abnormal gait, which was described as a heavy footprint, muscle weakness, and foot drop. By the time he was 14 years old, he had a major decline in his muscle strength in the lower body. Again, the upper body was fine. And by 15 years, there were some other tests that were being run, EMGs and NCB tests, which were indicating that he had a probable sensory motor neuropathy, demyelinating type. He had normal CBK and normal MRI tests. At age 15, he had his first genetic test, which was a Charcot-Marie tooth panel that was negative. At 16, he had an MRI that was negative. At 17, he had a negative 112 gene comprehensive neuropathy panel. And at age 17, he was negative for a hereditary spastic paraplegia comprehensive panel. So this is, again, an example of multiple tests. Each one of these is going to have a turnaround time associated with it, and then you get a negative result, order another, order another, order another. And you don't have to do that. You can order one comprehensive test that would test all of these genes and then some. So for this patient, he was positive for Friedreich ataxia, which was caused by 2 pathogenic variant types. One was a sequence variant, and one was a repeat expansion. And about 4% of patients with Friedreich ataxia will have this combination of both a small sequence change and a repeat expansion. The remaining individuals will have 2 repeat expansions, or 2 expanded alleles, as they call them. So this was an interesting case because it did take quite some time, and there were a large number of tests that were run for the patient before they sent the sample to Variantyx, and we got the positive for them. Also, that none of the tests that were in the clinical notes said that they ordered an ataxia panel. So if they had thought he had ataxia, they would have maybe ordered an ataxia panel, and then got an answer maybe if the panel included small sequence changes, because sometimes they can just be focused on the repeat expansion. Okay, so we'll go to another case. This is a 39-year-old female with over a 25-year diagnostic odyssey. She had progressive gait and balance difficulties. This was juvenile onset for her. She has paraplegia, dysarthria, difficulty coordination of the upper extremities. And she had previous testing. She had 2 tests for Friedreich ataxia. The clinician really thought this was Friedreich ataxia. She had the repeat expansion test. Like I said, sometimes the test is just for the repeat expansions because that is the most common result for a patient with Friedreich ataxia. So she was shown to have 1 expanded allele on that test, but she could just be a carrier. They didn't find a second pathogenic variant, and Friedreich ataxia is a recessive disorder, and you need 2 pathogenic changes. 1 is insufficient to cause disease. And so then they ordered a comprehensive ataxia panel that had repeat expansions and gene sequencing, and that was negative as well. They did pick up the expanded repeat expansion in the FXN gene, but they still reported her as a carrier. And at this point, the clinician was starting to think maybe it's not Friedreich ataxia, you know, 2 negative Friedreich test results saying carrier, but that could be a coincidence. And started to think maybe she has mitochondrial or something else. So ordered a broad test, the genome test from Variantyx, to try to see if there was something else causing her phenotype. But as it turns out, the clinician was right. She did have Friedreich ataxia, but she had a variant type that's very challenging to detect or impossible to detect on the current assays that are utilized today, typically for diagnosis of Friedreich ataxia. So she had a 2.7 kilobase deletion, so that's that structural variant here. And then she had a repeat expansion that the other assays had identified as well. So we were able to give a definitive diagnosis to the clinician, which is what he expected, suspected all along. But this size of a deletion, which is around the exon level, just 1 exon, is challenging to pick up on even next generation sequencing exome tests, because when I was showing you earlier, if you didn't, when you were not able to get all of the exons sequenced with really even coverage, then a small deletion may just look like noise, and so it won't really look like a true deletion. So there's size limits as to how big the structural variants need to be in order to be detected on those assays that don't do full genome sequencing. So that's why that one got missed by the other labs. Our next case is a 73-year-old woman with a over 12-year diagnostic odyssey. She has progressive cerebellar ataxia, progressive paraparesis, slurred speech, loss of feeling in her feet, and her onset was in her 60s. And for her, we also had a positive for Friedreich ataxia, but this is a very late onset Friedreich ataxia phenotype, which was consistent with the findings. So she had 2 expanded alleles, but one of them was expanded to about 100 repeats, and the other one was expanded to about 1,000 repeats. And when you have 2 expanded alleles of different sizes, the phenotype that is consistent with that finding is a very late onset form of Friedreich ataxia, which is what this woman has. So that is just one set of cases that sort of talks about why it's very challenging to identify patients with ataxia, especially this recessive ataxia that requires you to be able to identify multiple variant types. And the use of whole genome testing really does uniquely allow us to identify homozygous repeat expansions, which is what's typical for Friedreich ataxia, but also compound heterozygous changes, which means one chromosome has one type of change and the other chromosome has another. So they can have a repeat on one of the chromosomes and a small sequence change on the other, or a deletion duplication on the other. And the laboratories other laboratories have a big challenge with trying to identify that deletion duplication, especially if it's of a smaller size. So we can talk a little bit more about some other genes other than FXN. Variantyx does test 12 different ataxia-associated genes that have repeat expansions associated with them. So that allows us to do the comprehensive ataxia testing that we do, which includes not only those 12 genes that cause ataxia by repetitive expansions, but also all the genes that are associated with ataxia that may be caused by things other than repeat expansions. So we can go into another case. This is a 37-year-old male. He has progressive gait balance difficulties, pontocerebellar atrophy, and an abnormal MRI. And what we identified for this individual was a repeat in a CAG repeat in the ataxin-2 gene, which causes spinocerebellar ataxia type 2. We'll look through another case, 44-year-old male, 8-year diagnostic odyssey, progressive gait balance difficulties, stiffness with dystonia and dysarthria, difficulty with fine motor movements, and his onset was in his 30s. He had multiple tests done before they sent the test to us. He had negative mitochondrial tests, negative single gene analyses, negative exomes, negative repeat exome, negative repeat expansion test. Negative repeat exome analysis. Hopefully he didn't have a negative repeat expansion test, because he was positive for 2 expanded alleles in the Ataxin 8OS gene that is consistent with a diagnosis of SCA8. And it's known that for this particular ataxia that having both of your chromosomes, even though this is a dominant disorder, in one expansion is sufficient to cause disease, that the penetrance is much higher for individuals who have 2 expanded alleles. So the next case is a 60-year-old female with a 10-year diagnostic odyssey with progressive gait balance difficulties, mild dysarthria, mild dysphagia, and onset in her 40s. She also had 2 expanded alleles in Ataxin 8OS for a diagnosis of SCA8. And then another 37-year-old female with over a 5-year diagnostic odyssey with familial axonal neuropathy, asymmetric leg weakness, no associated pain, numbness, or tingling, and her onset was in her 30s. She was positive for a 6 base repeat expansion in the NOP56 gene, and that NOP56 is associated with Spinocerebellar ataxia type 36. So she had 1 expanded allele, which is sufficient for disease in this case. So what kind of the take home from that is that Variantyx tests will uniquely identify all variant types in all ataxia-related genes for your comprehensive ataxia testing. And we can go through another case here. This patient has ataxia, dysphagia, memory impairment, Parkinsonism, hypertonia, and speech articulation difficulties. And what we found here, so even though ataxia is part of the phenotype, we had a positive in something that's not really an ataxia disorder, but it's a prion disease. So he was positive for pathogenic a sequence variant in PRNP, which causes Gerstmann-Straussler-Schleicher disease. And this gene would not typically be included in ataxia panel, and it's not due to a repeat expansion. So again, a broad comprehensive test can really identify and distinguish, hereditary ataxia disorders from disorders that have ataxia as part of the clinical clinical spectrum of disease. In this case, we have a 21-year-old male, 2-year diagnostic odyssey, ataxia, speech difficulties, oculomotor apraxia, and nystagmus. He was positive for compound heterozygous pathogenic and likely pathogenic variants in SPG7s. This is recessive. Spastic dysarthria, dysphagia, pale optic disc, nystagmus, and cerebellar atrophy are reported in patients with this disorder. SPG7 would not be included in repeat expansion testing or in the ataxia panels, so the comprehensiveness of the test allows for phenotypes that may not be presenting as like a pure ataxia, but has ataxia as part of the clinical symptoms. To sum up, Variantyx can identify these difficult to detect causal variants in a wide variety of ataxia disorders, and even disorders that aren't really pure ataxia but may have ataxia as part of the clinical phenotype. Variantyx testing genomes, just kind of want to go back to that main platform again, just to reiterate that the purpose of doing full genome sequencing isn't necessarily to get all of that sequence information, but to create a backbone or a platform to be able to use software tools to identify other variant types. So in doing so, the genomes don't return like a lot of additional uncertain variants or deep intronic variants unless the variant in the intron is actually pathogenic and associated with disease, then it gets reported. They don't take more time to get a result, and they don't cost your patients any more than any other test. What they do is shorten the diagnostic odyssey. They detect all major variant types, so you can be sure that you would get a positive result for somebody who has any combination of variant types. Also, it's comprehensive, so if they have a confusing clinical phenotype where you're not sure if it's ataxia, all the other genes are also sequenced. So we really do have a test that is fit to the patient's clinical symptoms, not necessarily like any particular one diagnosis. They cost less than doing this stuff sequentially, and obviously with a shorter turnaround time, and provide the most comprehensive testing available. The other kind of benefit here is that you have the ability to run a reanalysis. So because we sequenced everything, as the patient's clinical symptoms may change or progress, or as variants are reclassified or new disease associations are connected to genes, all of these things can allow for a reanalysis of the data to see if there's a clinical match between a variant that's identified in a gene in your patient and the clinical symptoms of your patient. So that allows you to kind of requery the data set in the event that you have a negative test result, because as new information comes out, we could end up reanalyzing and getting a positive result later. So I want to thank you for your attention. If you want to learn more about whole genome sequencing and genomic unity and testing at Variantyx, you can come to our website at variantyx.com, or email us at info@variantyx.com, or call us at 617-209-2090. Thank you so much.